Ever wondered how scientists read the genetic code? Heard of Sanger DNA sequencing but found it too complex to understand? Well, let's break it down. DNA sequencing, as the name suggests, is a technique used to determine the precise order of nucleotides within a DNA molecule. Now, Sanger DNA sequencing, named after its developer Frederick Sanger, is a method that has revolutionized the world of genetics. It's been instrumental in mapping out the human genome, and has played a key role in the development of many of the genetic treatments and therapies we see today. But it's not all high-tech labs and white coats. It's a process that, once understood, can be appreciated for its elegance and simplicity. Common misconceptions might lead you to believe it's only for the elite scientists or that it's too complex to grasp. Well, worry not. Today we demystify the Sanger DNA sequencing technique for you. At its core, Sanger DNA sequencing is a method to read the genetic code. Now, what does that mean exactly? Well, let's break it down. DNA sequencing, in the broadest sense, is the process of determining the precise order of nucleotides within a DNA molecule. Nucleotides, for those who might not know, are the building blocks of DNA, consisting of a nitrogenous base, a sugar, and a phosphate group. In the context of genetics, these nucleotides are represented by 4 letters: A, C, G, and T, each standing for a specific base: adenine, cytosine, guanine, and thymine respectively. The unique sequence of these 4 bases in a DNA molecule is what encodes our genetic information. Think of it as a 4-letter alphabet that spells out the words and sentences of our genetic code. So where does Sanger DNA sequencing come in? Named after its developer, Frederick Sanger, this technique is one of the most commonly used methods for DNA sequencing. It's like a magnifying glass that allows us to read the genetic code. The purpose of Sanger DNA sequencing is to determine the exact order of nucleotides in a particular DNA molecule. This is crucial in many areas of biological research, including genomics, evolutionary biology, and medical diagnostics, among others. It's through this method that we're able to identify genetic variations and mutations that could lead to disease or give insights into our evolutionary history. In essence, Sanger DNA sequencing is the key to unlocking the secrets hidden within our DNA. It's a tool that helps us to translate the complex language of our genetic code into something that we can understand and interpret. By unveiling the precise order of nucleotides, it lays bare the intricate blueprint of life. So, it's like deciphering the secret language of life itself. Now let's dive into the process of Sanger DNA sequencing. Imagine the process as a journey with 4 main stops. First up, we have DNA extraction. This is where we obtain our DNA sample. It's a bit like fishing, but instead of fish, we're after the genetic material. We isolate the DNA from the rest of the cell components, giving us a clean sample to work with. Next, we arrive at the amplification station. Here we use a technique called polymerase chain reaction, or PCR for short. Think of it like a photocopy machine for DNA. PCR makes millions of copies of the specific DNA segment we're interested in. This ensures we have plenty of DNA for the next step. The third stop on our journey is the sequencing reaction. This is where the magic of Sanger sequencing really happens. We add a special mix of ingredients to our DNA sample, including DNA polymerase, a type of enzyme that helps to build DNA molecules and nucleotides, the building blocks of DNA. But here's the twist. Some of these nucleotides are specially marked. When one of these marked nucleotides is incorporated into the DNA strand, the building process stops. This creates DNA fragments of varying lengths, each ending with a marked nucleotide. The final stop is analysis. We separate these DNA fragments by size using a technique called gel electrophoresis. This allows us to read the sequence of marked nucleotides, which in turn gives us the sequence of our original DNA sample. It's like reading a book, but instead of letters, we're reading nucleotides. And that's how we unravel the genetic code, one nucleotide at a time. The Sanger sequencing process may seem complex at first, but once you break it down into these 4 steps: extraction, amplification, sequencing reaction, and analysis, it becomes much more manageable. Just remember, like any journey, it's one step at a time. Now that we've understood the process, let's tackle some common misconceptions and difficulties. First and foremost, there's a widespread belief that Sanger DNA sequencing is an overwhelmingly complex process. While it's true that the technique involves intricate steps, it's important to remember that complexity doesn't equate to unlearnability. In reality, each step of the process serves a specific purpose and follows a logical order. So, if you feel daunted, take a deep breath and approach each step individually. Another common misconception is that Sanger DNA sequencing requires expensive, high-tech equipment. While it's true that certain tools and machines are necessary, they are not as out of reach as you might think. Many laboratories around the world use this technique, and it's not exclusive to high-budget facilities. In fact, the key ingredients for Sanger sequencing, the DNA polymerase, primers, and nucleotides, are used in many other biological procedures as well. Now let's talk about some difficulties in understanding the process. One major challenge can be the technical jargon often used to describe it. Words like polymerase, deoxynucleotide, and capillary electrophoresis can be quite intimidating. But don't let this discourage you. Each of these terms describes a specific part of the process and, once understood, can make the overall picture much clearer. Another hurdle can be visualizing the process. It's not always easy to imagine what's happening at the molecular level, but remember, there are plenty of resources available to help. Diagrams, animations, and models can all bring the process to life. Lastly, some might find the interpretation of sequencing results challenging. Reading the colored peaks on an electropherogram can seem like deciphering a secret code. But with practice and understanding of the process, it becomes much simpler. So don't be disheartened by these misconceptions and difficulties. Remember, understanding any scientific process is about breaking it down into manageable chunks and taking one step at a time. To wrap up, here are some handy tips and tricks to help you understand Sanger DNA sequencing better. Firstly, it's important to visualize the process. Picture yourself in a lab, surrounded by test tubes and pipettes. You're not just memorizing steps, you're conducting a symphony of science. Each step in the process has a purpose, and this visualization can help you remember what happens and why. Next, try associating each step with a simple analogy. For example, you could think of the DNA as a long, complicated recipe. The goal is to read this recipe, but it's written in a language we don't understand. The Sanger method is like a translator, helping us understand each ingredient and cooking instruction. It's a translator that works letter by letter, translating the DNA language into something we can understand. It's also useful to remember the purpose of each step in the process. This can help you understand why things are done in a certain order and why certain materials are used. For instance, consider the role of the DNA polymerase enzyme. It's not just a random ingredient, it's the chef that follows the recipe. It reads the DNA and assembles the new strand. Understanding its role can help you remember why it's important in the process. And don't forget about the terminating dideoxynucleotides. These are like full stops in our DNA sentences. They signal the end of a sequence and allow us to read the DNA one letter at a time. Lastly, practice makes perfect. Try explaining the process to someone else, or write it down from memory. You could even draw a diagram or create a flowchart to help you visualize the steps. The more you engage with the information, the easier it will be to remember. With these tips and a bit of practice, you'll be decoding the secrets of DNA in no time. And there you have it, a simple guide to understanding Sanger DNA sequencing. We've journeyed from the basics of what this technique is, all the way through to the nitty-gritty of the process. We've addressed common misconceptions and difficulties, and armed you with tips and tricks to help you navigate your way through the complex world of Sanger DNA sequencing. Remember, this technique is a powerful tool in the field of biology, allowing scientists to read the genetic code that forms the blueprint of life. It's an intricate dance of nucleotides, a symphony of A's, T's, C's, and G's that tell the story of who we are on a molecular level. The importance of Sanger DNA sequencing cannot be overstated. It is the key to unlocking the mysteries of the genetic code, and with a good understanding of the process, you're well on your way to cracking that code. So next time you hear about DNA sequencing, remember, it's not a secret language, it's a code that you too can crack.